Hello, thank you for joining us today as we'll talk about how the Levi cell from our company LevitasBio can help you really get the most out of your sequencing workflows. As most of you are probably aware, the actual quality of the sample going into the sequencing reaction is the most fundamental and most important part of any workflow. The reason this step is so important is because any changes in the gene expression profile, cell state, or cell health will not be able to be corrected in a downstream analysis, but will be carried through the entire process. As the old saying says, garbage in, garbage out, unless you're really able to start with the highest quality sample in terms of both cell viability and any sort of cell damage, you will not get the best quality data. And quite often, the results will actually not be biologically relevant. And this is the part of the workflow that we at LevitasBio focus on. Regardless of your starting cell type, whether it's blood, dissociated tissue, or even ag-bio samples, we are able to provide a solution to the most critical bottleneck in most of these sequencing processes. Our solution allows you to get the most out of your sample in a fast and simple fashion. And most importantly, from our work with our customers and internal data, we have been able to demonstrate over and over again that the quality and the gene expression profiles we generate are as true to what is occurring in nature or what is true as possible. Now, as you may have heard, our company is primarily based on a label-free sample enrichment solution. What this means is that we don't actually use any labels, dyes, or antibodies to identify the viable cells of interest. And this is really one of the fundamental benefits of our technology. Since the cells are not modified in any way, shape, or form, that means they are not altered, they're not perturbed, and we're able to provide these key biological insights. Now, as a company, we're actually quite young. We were established in 2017, but the fundamental work on which we are based goes back to almost a decade prior. The original inventor is George Whitesides from Harvard University, and his goal was to be able to develop a technology that would allow you to isolate and characterize particles without any labels whatsoever. We licensed this technology in 2017, started an early access program in 2018, and have released our first commercial system, the Levi cell, in the summer of 2020. With over a year of cells under our belt, we've learned quite a bit about this market, and we've really been able to accelerate our product development that we'll talk about a little bit later on. Our team has worked together from the beginning, and in the last year alone, we've been able to be rewarded with 4 new innovation awards for the work we've done on this technology. Now, as I've mentioned in the past, one of the key aspects of any workflow is to be able to produce data that is meaningful, that does not change the understanding of biology, that improves it. And so what we have developed is this method that effectively allows you to see nature really the way it's meant to be. And the same method, same technology, same approach works for all applications, whether it's associated tumors, blood, primary samples, or really we're quite open to any sample type as a starting sample source. Now, as you know, we've been around for about 4 years. And over that time, we focused on really validating the technology and showing its utility in isolating viable cells or cells of interest in everything from blood to biopsies. Now, over the last year, we've really improved this workflow by incorporating several additional steps. We can remove dead cells, we can remove debris. We are working on a new scavenger product that removes RNA and DNA. This confounding information from RNA, for example, that's free floating in the solution in which the cells are, is quite often the most confounding part of any experiment. And again, this is a complete workflow that allows you to enrich for specific cells, whether it's a downstream sequencing reaction, as a upfront workflow for a flow cytometry reaction, or a CRISPR experiment where you're trying to enrich for transfected and edited cells. All of these workflows have already been validated, and our team has specific examples and customers that we can talk to you about. Now, what's really interesting about our technology is how scalable it is. It effectively allows us to work with any cell, any particle size. We've demonstrated technology to work down on a molecular level. This is what George Whiteside's lab originally worked on. And in fact, we now have customers that sort C. elegans, embryos, or organoids. So the size is a great enabler for us, and that the same platform, the same technology allows you to work with samples and particles of any size. The same is true for the technology in terms of how easy it is to use. We do not pre-filter. We don't have to calibrate anything. We use less than half a PSI of pressure. So this is not a microfluidic device. It is a macrofluidic device that really allows you to isolate any particle of interest without having to worry about background contaminating particles, any sort of pre-enrichment or cleanup afterwards. Now, the workflow could not be simpler. You effectively take your sample, you dispense it into one of our single-use cartridges. The cartridge goes into the LeviCell, and from there, within 20 minutes, with no additional hands-on, the LeviCell isolates your cells of interest without any dyes, without any antibodies. It has a full characterization capability, which means the sample is imaged and can be analyzed so that you obtain information such as how many live cells are in my sample. Is there a lot of debris? Is this quality going to be good enough to sequence? And from there, once you have that information, you can choose to collect the particles of interest and then move forward to a downstream sequencing reaction, a culture, whatever it is that you are going to use those cells for. Now, inside of the system, we have two rare earth magnets. These are traditional rare earth magnets, very similar to what you would place on your fridge. These are about 2 millimeters apart, and they create a very strong magnetic field. Our consumable effectively is placed between these two magnets. Now, this magnetic field is creating a very strong density gradient that we control. And the modality in which we're actually isolating the particles is based on this field. Now, while this is occurring, we are imaging the entire reaction in real time in both bright field and fluorescence so that you can incorporate dyes or markers if you choose to do so in order to optimize the experiment. And this recording is occurring in real time. So you're actually watching in real time as the particles are separating. Now, let's say we start with a dissociated tissue. This is a complex sample, and it will require several modes of operation that can all occur in the LeviCell at the same time in order to obtain the final sample of interest. Step number one, let's say it's viable cell enrichment. We want to make sure we enrich for all the live cells, getting rid of dead cells or small and large debris as a first step. Well, live cells levitate. We are able to basically enrich for the live population. Now, but we're not done yet. The second mode of operation will then allow you to go ahead and pull out these cells of interest where we are now remove the debris in the background RNA and DNA and collect them. The way we do that is inside the cartridge. The cartridge has two collection ports. Your cells of interest come up top, and the waste, everything else goes to the bottom. These cartridges are single use. They are fully irradiated for sterility. And unlike a microfluidic device, they can accommodate very large particles. As I mentioned, embryos, organoids, things up to 300 micron can easily be sorted here, right? Now, the key to the ease and utility of the system is that each of these cartridges fully contains the sample. The sample never leaves the cartridge. There is no cross-contamination possible between experiments. Basically, once you're done with the experiment, you take the cartridge out, you throw it away. There's no cleaning, there's no calibration, experiment to experiment or run to run. Now, in terms of workflow, because we have incorporated these multiple steps and no requirements for purification or pre-enrichment, our entire workflow is effectively 3 steps in about 20 minutes. This is a great improvement in both time, hands-on, and overall, versus a standard method such as fax or bead processing. And our really focus over the last few years was to develop a technology that allows you to get access to some of the most difficult and most precious samples. These are either samples that are very low viability, frozen samples, archive samples. We work with samples that are down to, say, 10% viable cells and can enrich them to over 90%. We also specialize in any sample that has very few cells. We've gone as few as few hundred viable cells and still have been able to extract those and process those quite sufficiently. And finally, the area that we are gaining the most ground and we hear the most from customers is on our ability to work with very difficult cell types, whether these are adipocytes, macrophages, pancreatic cells, cells that typically are very difficult to work with and cause many problems, are very easy to use. Again, we use half a PSI of pressure. There's no nozzles, there's no squeezing. It's a very gentle process that does not cause any gene expression changes or any perturbations on the cells themselves. Now, obviously, data kind of speaks volumes here. So you can see from this example, in terms of our viability and our ability to get a very high yield, whether we start with 1 million cells or 20,000 cells, we're able to achieve almost 100% viability and average of 80% yields on all different starting cell numbers, right? Unlike a traditional experiment where you're using a bead workflow or a fax instrument, where the efficiency plummets as you go down in cell number, for the Levi cell, it doesn't really care whether it's a few hundred cells or a few million. The performance is effectively the same. Now, obviously, one of the most important things that we've spoken about is the Levi cell's ability not to alter gene expression. And again, this is a very nice experiment with 3 different controls where a cell line is run across, is sequenced. And what we're looking for is to see if the control versus the sample process on the Levi cell has seen any gene expression changes. And effectively, what you're looking at is just noise. There is no significant gene expression in any of the detected genes, even on the sample that's sequenced to its full depth when processed in the Levi cell. This is very unique. Usually, you will see significant alteration in profiles. Now, we've done quite a bit of work on different cell types to show the performance. Again, on average, we'll see 100% viability or very close to 100%, and something with 70 and 80% yields. Now, when we compare that to workflows such as bead-based workflows from Miltenyi, the biggest improvement we see here is in yield, right? We have 2 different tumors here, a colorectal tumor and a bladder tumor, both starting around 200,000 cells. And effectively, what you're seeing, while the Levi cell generates quite a nice yield, almost 80% yield, 20,000 cells coming out of this, sufficient for a single cell workflow or sequencing reaction. The bead workflow, unfortunately, does not. There's simply not enough surviving or viable cells coming out of the workflow to really do anything with. Now, we've done the same kind of work validating our performance across many different cell types. These are primary cells, in addition to cell lines, things like cardiomyocytes, very difficult to work with macrophages, things that are easily activated or damaged. And we're running the same sample, Levi cell versus fax. What do we see with very few cell numbers? The Levi cell outperforms the fax workflow in terms of both viability and yield. And again, this is mostly due to the fact there's no label, there's no high pressure, there's no squeezing. The cell's not stressed in any way, shape, or form. Now, obviously, one of the nicest examples we could show you here is an actual single cell workflow. This is a single cell workflow with a 10x sequencing platform. We're starting with about 400,000 cells. This is a primary tumor sample. This work was performed by Discovery Life Sciences. And we're comparing a traditional protocol they use to a Levi cell enrichment. Now, the key in this data is that even though we have 2 different samples processed in 2 different ways, we started with the same number of viable cells, about 400,000. We split the sample into 2, and both the Levi cell sample and the standard method were sequenced using the same depth. So the same number of sequencing reads, same number of barcodes are detected. So the differences in data you see here are not due to sequencing deeper. They are because effectively we are seeing more genes. So in both of these samples, early stage and late stage, the Levi cell workflow not only showing many more viable cells, but more importantly, within those cells, we're detecting up to 3 times up to 6 times more unique genes, which is hugely powerful when we actually look at how the gene expression shows up. The conventional sample is clearly lacking not only genes, but also expression profile. If we look at the LeviCell results, you're seeing 6 times as many genes and 10 times as many unique mRNA molecules. So in summary, what we've developed over the last few years is a very simple, very comprehensive method that allows you to get the most out of your sample, not only in terms of yield and viability, but more important, in terms of providing you the gene expression information, providing you the biological insight that is only possible when processing with a simple method that does not alter or does not perturb the cell or the samples. With that, thank you for your time, and I'll take a few questions here. Looks like one of the questions is, can the system replace fax? Well, in some of the workflows, it certainly can. We also have customers that are actually using the LeviCell up front of a fax. The idea is that they have very difficult to work with samples, very dirty, lots of debris, lots of contaminants. And they actually use the LeviCell to pre-enrich or pre-clean the sample before going on a fax. So it can be a replacement, but you can also use it in conjunction with a fax system. See, the next question we hear here is, how many cells can I use with? On the lower end, we really recommend you stick to at least 5,000 to 10,000 cells. We have gone as few as few hundred. Obviously, the fewer the cells, the more difficult it is to even quantitate the output. On the max number, anywhere from 1 to 2 million on average is a good number to start off with. Depending on the cell size, you can load more cells or fewer cells. Bigger cells take up more space, so obviously we can't load quite as many of those. And then the other question we have here is, are there other applications? Well, we have several new applications. We have a new tissue dissociation kit that's coming out that allows you to start directly with tissue, and it's optimized for the LeviCell workflow. We are also incorporating cell surface marker depletion kits. So for things like CD45, if you're trying to deplete those cells along with live cell enrichment, those kits are coming out. And so if you'd like to learn more about those or join our early access program, please let us know. With that, I thank you very much, and have a great day.